lecture notes on the Google? Yes? You have a problem? I would like you to know in general, very deep, especially in the biochemistry. That's very important. But for cell biology class, you will not be asked questions to reproduce the entire TCA cycle in any questions. Not only that, none of the structures you need to be reproduced in your right? Because I'm sure that
for biochemistry, I'm going to be studying more this semester.
we will be asked about the TCA cycle in any okay? So other than for you to understand where these electrons are coming from, which eventually will be used in the electron transmission, right? They're coming from the reduced equivalents, NADH2, and those are forming the TCA cycle. That will be enough for cell biology. Okay? Any other questions? No. Okay, so let's today talk about, continue talking about mitochondria, specifically about this transfer of electrons. And electrons, the transfer is a driving force for the formation of ATP, for the synthesis of ATP. And those of you who took biochemistry 1 already by that time, I guess it's correct with it, right? So probably most of you, or maybe you already perhaps know about that. So let's go through this briefly. So the important point here, yeah, the transfer of electrons through so-called electron transfer chain. This is different components of this electron transfer chain. The transfer of electrons eventually will lead to the synthesis of ATP, this universal metabolic fuel that will be used in many different reactions, not only in mitochondria, but outside mitochondria as well. Okay? Now, the electrons shown here, they originate from this reduced equivalent. The reduced equivalent being NADH2, NADH, nicotinamide dinucleotide reduced, right? And FADH2. Now, both kind of reduced equivalent, NADH and FADH2, obviously are formed in the TCA cycle, Krebs cycle. In those dehydrogenase reactions, pyruvate dehydrogenase, alpha-ketoglycerol dehydrogenase, isocitrate dehydrogenase, and so on and so on. All right? And so as soon as they have been formed, this reduced equivalent can be now used by this electron transfer chain in order to get some electrons from these reduced compounds. And say, for instance, one of them, NADH, would dissociate forming NAD+. So it's positively charged molecule of nicotinamide dinucleotide, right? And the product of this dissociation would be NADH dissociate into NAD+, and the other product will be a proton, right? The proton, which is that point, stay in the matrix of mitochondria. So that's the matrix, that's intermediate space between the outer and inner membrane of mitochondria, right? Now, so now from the electrically neutral molecule, two positively charged molecules, or ions in this case already, they becoming ions are formed. So what is the other product? There are two other products of this reaction. Guys, ATP, no. Electrons, good. And obviously electrons charged negatively, right? So that would create a balance, would give you a balance. Two electrons have been formed, right? So this reaction of dissociating NADH is a spontaneous reaction, as any chemical reaction. And the only thing would it be still spontaneous, unassisted by a protein or a group of proteins that will be extremely slow, too slow to use these electrons in order to synthesize a significant amount of ATP eventually, right? So therefore there is an enzyme, or more exactly, this is a protein of complexes. And therefore this complex is formed protein complex number 1, right? And this protein complex number 1 also have name NADH dehydrogenase, or complex 1 of the electron transfer chain. not only accelerates the formation of this NAD plus and protons, right? Not only greatly associate by shifting equilibrium in this reaction towards the formation of NAD and protons, but also use these two electrons formed in this reaction in order to pump them move them from the matrix of mitochondria into this intermembrane space, right? So during this dissociation reaction, a certain amount of energy is released, which is used by this complex one of the electron transfer chain in order to move two electrons to the next component of this electron transfer chain. And the component marked here by Q, by the letter Q, is ubiquinone, or coenzyme Q. So it's a lipid-soluble molecule. It's basically a molecule of a lipid nature, which accepts these two electrons from complex 1. Now, in addition to being able to transfer these electrons to ubiquinone, complex 1 does something else. It's also moved the protons. The protons from the matrix, remember the proton has been formed already in the matrix in this reaction, right? So at this point there was a certain amount of protons. They obviously always are in the matrix. And so this complex 1 uses the energy which is produced during such transfer of electrons in order to move also protons from the matrix of mitochondria into intermediate space. And the importance of this movement of electrons and protons will become understandable a little bit later today. Now, in addition to that, the ubiquinone also getting two electrons from another complex 2. And the complex 2 of the electron transfer chain is an enzyme of the TCA site, the succinate dehydrogenase enzyme, the one which is synthesized fumarate from succinate in this Krebs site, right? And the product of this reaction is FADH2, adenine dinucleotide reduced. And that FADH2 serves as a source of two other electrons which are also transferred from the complex 2 to ubiquinone. It's the same acceptor of electrons as the one which accepts electrons from complex 1. Now, the difference between these two complexes, two electron transfer chain complexes, the ETC complexes, is that one of them, this complex 1, is a transmembrane protein. It's crossed the entire inner membrane of mitochondria. And therefore, that enabled this protein complex to have a certain type of channel through which protons can be moved from the matrix to the intermembrane space. Now, complex 2, succinate dehydrogenase, is not a transmembrane protein. It does not cross the entire inner membrane. So therefore, this complex 2 is unable to transfer any protons. Just transfer electrons to ubiquinone, but not pumping protons into intermediate space, because it's physically unable to do so. It doesn't cross the membrane. All right? So now, from this ubiquinone, this lipid molecule, accepting two pairs of electrons, one from complex 1, another pair from complex 2, ubiquinone transfer electrons to the next component of this electron transfer chain, so-called complex 3, right? And also called complex BC, cytochrome BC1. And it's called BC1 complex because these two cytochromes are the major component, protein component of this complex 3. But again, when we talk about complex, we assume that this is really complex, when many proteins come together, cytochrome B, cytochrome C1, and many other proteins in order to form this complex, right? So not only complex 3 would take electrons from ubiquinone and transfer a pair of electrons to the next component of electron transfer chain, cytochrome C, but it will also, the complex 3, would also pump protons across the inner mitochondrial membrane, taking protons in the matrix and moving them into the intermembrane space. All right? And again, complex 3, unlike complex 2, but like complex 1, is also a transmembrane protein. And that's the reason why it's able to transfer electrons, because inside of the body of this complex, there is a certain type of small channel through which protons can be moved from one space, from the matrix, to the intermediate space, intermembrane space. All right? Now, the next component of the complex is cytochrome C. So it's a small protein, molecular weight about 13 to 15 kilodaltons, smallest compared to most of the other proteins. And this protein, cytochrome C, is attached to the inner membrane by a certain lipid molecule. The lipid molecule which covalently attached to cytochrome C, and it's used as an anchor. Cytochrome C is this lipid molecule to be anchored, attached to the inner membrane. But it's extremely mobile, this cytochrome C. So it's kind of taking 2 electrons from complex 3 and then sliding on the surface of this inner membrane towards the complex 4. Eventually meeting complex 4 and transferring this pair of electrons to complex 4. All right? And so complex 4 by itself does the very terminal reaction of electron transfer. Right? All these previous complexes and protein and lipid components, cytochrome C being protein component and ubiquinone being a lipid component, they all were transferring electrons. Finally, the complex 4 would transfer these 2 electrons on molecular oxygen, and in doing so will produce a molecule of water. Right? And so because these complex, and the name of this complex is complex 4. Because it does a very specific chemical reaction, it transfers electrons to molecular oxygen and oxidize cytochrome C. It's called cytochrome C oxidase. Again, complex 4, just like complex 3 and complex 1, is a transmembrane protein. Cross the entire inner membrane, there's a certain type of channel inside through which, again, complex 4 use the energy of this electron transfer also to move protons from the matrix to the intermembrane space. All right? So at that point, when the complex 4 already transferred all this electron to molecular oxygen, water has been formed. You have a situation when this intermembrane space is extremely enriched in protons, hydrogen ions, right? Meaning that what would be, you say, the pH in the intermembrane space as compared to the matrix? Higher or lower? Lower pH, higher concentration of protons. So let's lead this proton transfer into acidification, slight acidification of the intermediate space. Now, when these protons have been moved by complex 1, 3, and 4, the gradient of protons is established. And this gradient is called electrochemical gradient, right? So it's definitely electrical potential, because there is a difference in the concentration between of positively charged ions, protons, between the intermembrane space and the matrix, right? And it's also chemical gradient, because there's a certain type of chemical, a proton or hydrogen atom is concentrated on one side of this inner membrane, in the intermediate space. So any kind of chemical gradient or electrical gradient, if you do something special in order to decrease this gradient, decrease the concentration of protons in this intermediate space, by, for instance, moving them back to the matrix, would create a certain type of energy. Just because from the thermodynamical point of view, if someone, like Maxwell Diamond, for instance, moving protons from one compartment to another, there is a certain type of energy released at that point, because it's movement down of the concentration gradient. So this amount of energy which produced during such a movement back of the protons from the intermediate space to the matrix can be used to drive a certain type of chemical reaction. And this reaction is the synthesis of ATP. So there is an enzyme which is called ATP synthase. And so it's a transmembrane protein, so it definitely can move these protons from one space, from intermediate space to another space, to the matrix, right? So there is a certain type of hole or channel inside of the body of this ATP synthase. And also called complex 5, just because it's called complex 4 in this order of events. And so by moving these protons from the region with high concentration of protons to the region with a small concentration of protons, a certain amount of energy is produced, and the